Inside your skull, there's about 3 pounds of tissue, roughly the weight of a cantaloupe, and it's using something like 20 watts of power. That's less than the dim bulb in your refrigerator. And yet, this little 3-pound thing is, as far as we know, the single most complex object in the observable universe. Not the most powerful, not the largest, not the fastest, the most complex. More connections inside it than there are stars in the Milky Way. And the really wild part? It built itself. No one designed it. No engineer sat down and drew up the plans. It emerged, slowly, over hundreds of millions of years, through trial and error, on an almost incomprehensible scale. And the story of how that happened, how a universe of dumb matter eventually produced something that could wonder about itself. Well, that's what we're going to talk about tonight. Let's start way back. And I mean way back, not thousands of years, not millions. We're talking about roughly 3.5 to 4 billion years ago, when the Earth itself was barely cooled down enough to have liquid water on its surface. The planet was maybe 700 million years old. There were no continents the way you'd recognize them. No oxygen in the atmosphere to speak of, just rock and water, and volcanic gas and ultraviolet radiation pouring down from a sun that was actually a bit dimmer than it is today. And somewhere in that hostile, churning world, maybe near a hydrothermal vent on the ocean floor, maybe in a shallow tidal pool, maybe in some crack in a mineral-rich rock face, chemistry crossed a line. Molecules started copying themselves. Life began. Now, these earliest living things were unimaginably simple. Single cells, no nucleus even, just little bags of chemistry, what we'd call prokaryotes. Think of something like a modern bacterium, though even that comparison is generous, because modern bacteria have had 4 billion years of evolution to get good at what they do. These ancient cells were simpler still. And here's the thing that matters for our story. They had no brains, no nervous system, no neurons, nothing remotely like thought or awareness. They didn't need any of that. They survived by responding to their immediate chemical environment through direct molecular interactions. If there was more sugar over here, they'd drift toward it. If something toxic was over there, they'd drift away. All of this was handled by proteins embedded in their cell membranes, receptors that could detect specific molecules and trigger internal chemical cascades that would, say, change the way the cell's little flagellum rotated, steering it in a new direction. And that, believe it or not, is where our story begins. Because that simple ability, sensing something in the environment and responding to it, that's the seed. That's the ancestor of everything your brain does right now. Every thought you've ever had, every memory, every dream, every moment of falling in love or solving a math problem or catching a ball thrown to you across a yard, all of it traces back to this one basic trick that a single-celled organism figured out billions of years ago. Detect a signal, process it, respond. That's it. That's the template. For a very long time, and I want you to really sit with how long this is. For roughly 2 billion years, life on Earth stayed single-celled. 2 billion years. That's almost half the entire age of the planet. If you compressed all of Earth's history into a single 24-hour day, multicellular life wouldn't show up until sometime around 6 or 7 in the evening. So for most of the story of life, there were no animals, no plants, no fungi as we'd recognize them. Just single cells, floating, dividing, competing, cooperating in simple ways, and slowly getting more sophisticated in their internal machinery. One of the big leaps during this long single-celled era, and this is genuinely one of those most important events in the history of life, was the evolution of the eukaryotic cell. That's the kind of cell you're made of. Unlike those early prokaryotes, eukaryotic cells have a nucleus, a little compartment where the DNA is kept. They have internal structures called organelles, mitochondria for energy production, and in plant cells, chloroplasts for photosynthesis. And the best current theory for how eukaryotic cells came to be is one of the most beautiful ideas in all of biology. It's called endosymbiosis, and it was championed by a biologist named Lynn Margulis back in the late 1960s, at a time when most of the scientific establishment thought she was, frankly, wrong. Margulis proposed that mitochondria, those little power plants inside your cells, were originally free-living bacteria. At some point, a larger cell engulfed a smaller one, but instead of digesting it, the two struck up a partnership. The smaller cell was really good at using oxygen to generate energy. The larger cell provided protection and raw materials. Over time, the smaller cell lost its independence. Its genes migrated partly into the host cell's nucleus, and the two became inseparable. One organism, and the evidence for this is now overwhelming. Mitochondria have their own DNA, their own double membranes. They replicate somewhat independently inside your cells. They're the descendants of ancient bacteria that moved in and never left. Why does this matter for the brain? Because eukaryotic cells, with their mitochondria, could generate far more energy than prokaryotes. And energy is the currency of complexity. You can't build a big, complicated organism, certainly not one with a brain, if your cells can't produce enough fuel. The evolution of the eukaryotic cell was like upgrading from a campfire to a power station. It didn't create the brain, but it made the brain possible. So now we're somewhere around 1.5 to 2 billion years ago, and eukaryotic cells are becoming more common, and eventually, some of them start doing something new. They start sticking together, forming colonies, cooperating, and this is where things get really interesting for us. Because the moment cells start living together in groups, they face a new problem: communication. In a single-celled organism, everything is local. The cell detects what's right next to it and responds. But in a colony, in a multicellular body, cells on one side of the organism need to know what's happening on the other side. If food is detected at the front, the back needs to know to keep moving forward. If danger is detected on the left, the right side needs to coordinate an escape. The earliest multicellular organisms solved this with chemical signaling. One cell releases a molecule. It diffuses through the surrounding fluid. Neighboring cells detect it and respond. This works fine when you're small. When you're a little blob of a few hundred or a few thousand cells, but chemical diffusion is slow, really slow. If you're a millimeter across, a chemical signal might reach the other side of your body in a reasonable time. But if you're a centimeter across, or 10 centimeters, diffusion alone starts to feel like sending a letter by horse when you need to make a phone call. And this is the pressure, the evolutionary pressure, that eventually gave rise to the neuron, the need for speed, the need to send a signal from one end of a body to the other quickly enough to actually do something useful with the information. And the solution that evolution stumbled upon was, honestly, kind of brilliant. Instead of relying on molecules slowly drifting through fluid, what if you had a specialized cell, a long, thin cell, that could pass a signal along its own length using electrical impulses? Not electricity the way a wire carries it, but something more subtle. An electrochemical wave, a chain reaction of charged particles, ions, flowing in and out of the cell membrane in sequence, like a stadium wave rippling around an arena. That's what a neuron is at its most basic. It's a cell that's been stretched out long and thin, and it can propagate a signal along its length at speeds that blow chemical diffusion out of the water. A chemical signal might travel through tissue at maybe a few micrometers per second. A nerve impulse can travel at speeds ranging from about 1 meter per second in the simplest systems to over 100 meters per second in the fastest neurons in your body right now. That's a difference of many orders of magnitude. It changes everything. Now, the earliest neurons didn't appear overnight, and pinpointing exactly when they first evolved is genuinely difficult, because soft tissue doesn't fossilize well. Neurons are squishy. They don't leave behind neat impressions in rock the way bones and shells do. So scientists have to piece the story together using a combination of comparative biology, looking at which living animals have neurons and which don't, and molecular clock analysis, which uses the rate of genetic mutation to estimate when different lineages diverged. And what this evidence tells us is fascinating. The most ancient animals alive today that have anything resembling a nervous system are the cnidarians. That's the group that includes jellyfish, sea anemones, and corals. These creatures have been around for at least 500 million years, and possibly closer to 700 million, and their nervous systems are beautifully simple. A jellyfish doesn't have a brain. It has what's called a nerve net, a diffuse web of interconnected neurons spread throughout its body with no central control point. When a jellyfish touches something with one of its tentacles, the signal spreads outward through the nerve net in all directions, like ripples on a pond, and the whole body responds. It can contract, it can pulse to swim, it can direct its tentacles toward prey. But there's no command center, no boss neuron telling everyone else what to do. And then there are the sponges, animals even more ancient than jellyfish, possibly diverging from the rest of the animal family tree over 600 million years ago. Sponges don't have neurons at all. No nerve cells, no nervous system. And yet, and here's what's really intriguing, sponges have genes that are remarkably similar to the genes that build synapses in your brain. Genes for ion channels, for neurotransmitter receptors, for the scaffolding proteins that hold a synapse together. The genetic toolkit for building a nervous system was apparently already present before neurons themselves existed. The parts were in the box before anyone assembled the machine. Think about what that means. It suggests that neurons weren't invented from scratch. They were cobbled together from pre-existing molecular components that were originally doing other jobs in the cell. sensing the environment, managing internal chemistry, communicating with neighboring cells. Evolution is a tinkerer, not an engineer. It doesn't design from a blank page. It repurposes what's already lying around. And in this case, what was lying around was a set of molecular tools that turned out to be spectacularly useful for building a rapid-fire communication system. So somewhere around 600 to 700 million years ago, give or take, in the murky depths of the Precambrian ocean, the first true neurons appeared. And once they did, animal life changed forever. Because now, for the first time, an organism could coordinate its body quickly. It could react to a predator in real time. It could pursue prey with something approaching intention. It could integrate information from different parts of its body. Touch here, chemical signal there, light over there, and produce a unified response. But a nerve net, like the kind a jellyfish has, can only get you so far. It's democratic in a way. Every neuron is roughly equal and signals spread everywhere at once. That's fine for simple behaviors, but it's not great for making decisions. If you want to do something more sophisticated, if you want to move toward food and away from danger at the same time and figure out which one takes priority, you need something more. You need neurons that are organized, clustered, concentrated in specific places where they can process information before passing instructions along. And that's exactly what happened next. In a group of animals that might seem humble to you now but were absolute revolutionaries in their time. We're talking about worms, flatworms specifically, and their relatives, the earliest bilaterally symmetrical animals, creatures with a front end and a back end, a left side and a right side. And at the front end, where the mouth was, where the animal encountered the world first as it moved forward, that's where the neurons started to cluster. That's where the first protobrains began to form. Little knots of nerve cells called ganglia, concentrated at the head end of the body, connected to simpler nerve cords running down the length of the animal. There's a flatworm you might have heard of, called planaria. It's tiny, maybe a centimeter or two long, and it's been a favorite of biologists for over a century because of its remarkable ability to regenerate. Cut one in half, and both halves grow back into complete worms. But what's really interesting for us is its nervous system. A planarian has a pair of cerebral ganglia at its head, two little clusters of neurons, connected by a bridge of nerve fibers, with two nerve cords extending backward through its body. It has simple eyes, just light-sensitive spots, really, connected to those ganglia. It can learn, not in any grand sense, but in simple conditioning experiments. Planarians can be trained to associate a stimulus with a response. They can form something that looks, if you squint, like a memory. And this humble little worm, or something very much like it, represents one of the most important transitions in the entire history of the brain. The transition from a decentralized nerve net to a centralized nervous system, from democracy to hierarchy, from signals spreading everywhere, to signals being routed to a specific processing center that could evaluate them, weigh them against each other, and issue commands. This is called cephalization, the concentration of neural tissue at the head end of the body. And it happened somewhere in the late Precambrian or early Cambrian period. roughly 550 to 600 million years ago. And what came next was an explosion. Not a metaphorical one. Well, actually, yes, a metaphorical one, but one so dramatic that scientists literally call it that, the Cambrian explosion. Starting around 541 million years ago, in a burst of evolutionary creativity that lasted maybe 20 to 25 million years. A blink of an eye in geological terms. Almost all the major animal body plans that exist today appeared in the fossil record. Arthropods, mollusks, chordates, echinoderms, the ancestors of insects and crabs and octopuses and fish and sea stars, all of them showing up in this relatively narrow window of time, and many of them for the first time had eyes, real eyes and teeth and claws and armor. Now, what triggered the Cambrian explosion is one of the great debates in evolutionary biology, and there are a lot of competing hypotheses. Rising oxygen levels, changes in ocean chemistry, the breakup of a supercontinent, the evolution of predation creating an arms race. Probably it was some combination of all of these. But here's what matters for our story. The Cambrian explosion was also a nervous system explosion, because once you have predators actively hunting prey, and prey actively evading predators, the pressure to process information quickly, to see, to react, to decide, becomes intense. Brains didn't just become useful, they became essential. And the animals that had better ones, the ones that could process sensory information faster and coordinate their bodies more precisely, those were the ones that survived to pass on their genes. There's a creature from the Cambrian that I think about a lot when I think about early brains. It's called Anomalocaris, and it was, for its time, an apex predator, about 1 meter long, which made it a giant by Cambrian standards, with large compound eyes, grasping appendages at the front of its head, and a circular mouth lined with plates. Its eyes alone had something like 16,000 individual lenses. To control those eyes, to coordinate those grasping arms, to hunt effectively in three-dimensional water, that required serious neural processing power. We don't have its brain preserved, of course, but we can infer from its behavior and its body plan that it had something substantially more sophisticated than a flatworm's paired ganglia. And around the same time, swimming alongside creatures like Anomalocaris, there were small, unassuming, worm-like animals that would turn out to be far more important to our particular story. They were soft-bodied, just a few centimeters long, and they had something running down their backs that no other animals had. A flexible rod called a notochord. These were the earliest chordates, the ancestors of every vertebrate alive today, the ancestors of fish, amphibians, reptiles, birds, mammals, the ancestors of you. And inside their simple bodies, running along that notochord, was a hollow nerve cord, not on the belly side, like in insects and worms, but on the back, a dorsal nerved cord, and at the front end of that cord, a slight swelling, a thickening, the faintest hint of what would eventually become a brain. Not a brain like yours, not even close, but the seed of one, the beginning of the beginning. And what's beautiful about this moment in evolutionary history is how humble it was. These early chordates, creatures like Pikaia, found in the Burgess Shale of British Columbia, or Haikuictus, discovered in the Chungjiang deposits of southern China, They weren't the dominant players. They weren't the top predators. They were small, they were soft, and they were probably near the bottom of the food chain. Pikaia was maybe 5 centimeters long, shaped a bit like a leaf, swimming with side-to-side undulations through Cambrian seas that were full of things that wanted to eat it. But it had that notochord, that dorsal nerve cord, and that tiny anterior swelling. And that swelling, that little concentration of neural tissue at the front end, that was the prototype for everything that would come after. Now, here's what's really important to understand about that swelling. It wasn't just more nerve cells packed into one spot. It was the beginning of a structural principle that would define vertebrate brain evolution for the next 500 million years. The idea of regionalization. Different parts of that swelling would gradually take on different jobs. Over vast stretches of time, that simple thickening would divide into distinct sections, each one specializing in processing a particular kind of information. And the earliest version of this division, the one that shows up in the most primitive vertebrate brains, is a 3-part plan: a forebrain, a midbrain, and a hindbrain. 3 bulges, 3 regions, 3 sets of responsibilities. Let's talk about that for a second, because this 3-part brain is one of the most conserved features in all of vertebrate biology. You have it. A fish has it. A frog has it. A bird has it. The basic ground plan was laid down so early and worked so well that evolution has never thrown it away. It's been modified, expanded, elaborated on in 1,000 different ways, but the fundamental architecture, forebrain, midbrain, hindbrain, that's been there since before anything with a backbone crawled onto land. In those earliest vertebrates, the hindbrain was probably the most developed of the 3. It sat closest to the spinal cord and handled the most basic functions: controlling the body's movements, regulating rhythmic processes, coordinating the simple motor patterns needed for swimming. Think of it as the operations center, the part that keeps the lights on. In your brain right now, the hindbrain includes structures like the medulla oblongata, which controls your breathing and heart rate, and the cerebellum, which coordinates your movements with an almost unbelievable precision. Those functions are ancient. They go all the way back. The midbrain, sitting in the middle, was primarily a sensory processing center. In early fish, it was dominated by a structure called the optic tectum, a region devoted to processing visual information. And this makes sense, right? Vision was becoming increasingly important. The Cambrian had kicked off an arms race in sensory systems, and being able to see, really see, not just detect light but form images, track movement, judge distance, that was a massive competitive advantage. The midbrain became the place where visual information was turned into action. See a predator, turn away. See prey, strike, fast, direct, sensory to motor processing. And then the forebrain. In those earliest vertebrates, the forebrain was the smallest and least impressive of the 3 regions. It was primarily devoted to processing smell, olfaction, which might seem like a humble beginning for the part of the brain that would eventually give rise to human consciousness. But think about what smell meant to a small, soft-bodied creature swimming through murky Cambrian waters. Smell told you where food was. Smell told you where predators were. Smell told you where potential mates were. It was the sense that connected you to the chemical landscape of your environment. And in a world where vision might be limited by turbidity or darkness, smell was often the most reliable source of information about what was out there. So that's your starting point. 3 regions, each with a primary job. Hindbrain for motor control and basic life support. Midbrain for vision and sensory motor integration. Forebrain for smell, and eventually for so much more. Now, the transition from these earliest chordates to true vertebrates. Animals with actual backbones, with skulls, with jaws. That happened over a long stretch of time, roughly between 500 and 400 million years ago. And during that transition, something remarkable happened to the brain. It got bigger, yes, but more importantly, it got more complex. New structures appeared, existing structures expanded, and a lot of this had to do with a fascinating genetic event, or rather a series of events that gave vertebrates a kind of genetic windfall. Here's what happened. At some point early in vertebrate evolution, the entire genome was duplicated. Not just one gene, not just a chromosome, but the whole thing, twice. 2 rounds of whole genome duplication. This is sometimes called the 2R hypothesis. And while the exact timing and details are still debated among geneticists, the basic idea is well supported. What it meant was that early vertebrates suddenly had extra copies of all their genes. And extra copies of genes are like spare rooms in a house. They give you space to experiment. One copy can keep doing the original job while the other is free to mutate, to drift, to potentially take on a new function. This is one of the major reasons vertebrates were able to evolve such complex brains. They had the genetic raw material to build new types of neurons, new signaling molecules, new brain structures, without losing the ones they already depended on. And you can see the results of this genetic bounty in the brains of the earliest jawed fish, which appeared around 420 million years ago. These were the placoderms and the early sharks and the ancestors of bony fish. And their brains were significantly more elaborate than anything that came before. The cerebellum, that hindbrain structure I mentioned, expanded dramatically in fish that needed precise motor control, especially in sharks, which are remarkable swimmers with an extraordinary sense of spatial awareness. A shark's cerebellum is, relative to its brain size, impressively large, and that makes sense when you think about what a shark does. It navigates 3-dimensional space. It tracks prey using multiple senses simultaneously. It makes rapid turns and strikes with precision. All of that coordination requires a sophisticated cerebellum. But here's where it gets really interesting. While the hindbrain and midbrain were doing their thing, the forebrain was quietly beginning its long, slow ascent to dominance. In early fish, the forebrain started developing a region called the pallium, a sheet of neural tissue on its outer surface. In fish, the pallium is relatively simple. It processes olfactory information, helps with some basic spatial learning, maybe contributes to simple decision-making. But the pallium is the evolutionary precursor to the cerebral cortex. Your cerebral cortex, that wrinkled, folded outer layer of your brain that handles everything from language to abstract thought, to the experience of listening to someone talk about brains. while you drift off to sleep. It all started as a thin sheet of smell-processing tissue in the forebrain of an ancient fish. And I think there's something genuinely wonderful about that. The most sophisticated structure in the known universe, the human cerebral cortex, began as a patch of cells devoted to figuring out what that chemical floating through the water might be. Smell, the most ancient, the most primal of the senses. And from that modest beginning, everything else grew. Now, while all of this was happening underwater, something else was brewing. Life was beginning to move onto land. Plants had already made the transition, and arthropods, insects, spiders, their relatives, were establishing themselves in terrestrial environments. And then, around 375 million years ago, give or take, certain lobe-finned fish began making the transition from water to land. This is one of the most dramatic transitions in the history of life, and it posed enormous challenges for the brain. Think about what changes when you leave the water. Gravity becomes a much bigger problem. You need to support your own body weight, which means your motor control systems need a complete overhaul. The sensory environment transforms. Sound travels differently in air than in water. Light behaves differently. Smell works differently. You're now detecting airborne molecules instead of waterborne ones. And the world is just more variable. Temperature fluctuates more on land. Humidity changes. The terrain is uneven and unpredictable in ways that an aquatic environment isn't. The fossil that captures this transition most beautifully is Tiktaalik, discovered in 2004 on Ellesmere Island in the Canadian Arctic by Neil Shubin and his colleagues. Tiktaalik lived about 375 million years ago, and it was a genuine intermediate between fish and the first tetrapods, the four-limbed vertebrates that would eventually give rise to amphibians, reptiles, and mammals. It had fins, but those fins had bones inside them that correspond to the bones in your wrist and arm. It had a flat head with eyes on top, like a crocodile, suggesting it spent time in shallow water looking up at the world above the surface. And its brain, while we can't examine the soft tissue directly, left impressions inside its skull that tell us something about its structure. The forebrain was beginning to expand. The regions devoted to processing airborne sensory information were growing. The brain was already adapting to a world it hadn't fully entered yet. And this is one of the patterns you see over and over in brain evolution. The brain doesn't wait for the body to finish changing and then catch up. The brain changes alongside the body, sometimes even leading the way. Neural circuits that would be useful in a new environment begin to develop, while the animal is still only partially committed to that environment. It's as if evolution is hedging its bets, building the software before the hardware is fully in place. The first true tetrapods, animals like Acanthostega and Ichthyostega from around 365 million years ago, had brains that were still broadly fish-like in their basic plan. 3 regions, same general layout. But the proportions were shifting. The forebrain was getting a little larger. The olfactory bulbs, the smell processing centers, were prominent, which makes sense because smell was becoming even more important on land, and the parts of the brain devoted to motor control were adapting to handle the completely new challenge of walking. of coordinating 4 limbs against gravity on an uneven surface. Here's something to think about. When you walk across a room, your brain is performing calculations that would challenge the most advanced robotics engineers alive today. The constant adjustments to balance, the coordination of dozens of muscles firing in precise sequences, the real-time processing of sensory feedback from your joints and muscles and inner ear. All of that had to be invented, neurologically speaking. When vertebrates moved onto land, fish didn't need it. Their bodies were supported by water. But once you're on land, every step is a controlled fall, and your brain has to manage that, millisecond by millisecond, without you even thinking about it. And that challenge, the challenge of terrestrial locomotion, drove significant changes in the cerebellum and in the motor cortex. These structures expanded and refined themselves over millions of years, and the early amphibians were the testing ground. They were clumsy by modern standards, probably. Their limbs splayed out to the sides. Their movements were likely slow and effortful, but they were walking, and their brains were learning, in an evolutionary sense, how to make that work. As the millions of years rolled on and amphibians diversified, some lineages began spending more and more time on land, and one group in particular began developing a feature that would change everything. Not just for brains, but for life on Earth. They started laying eggs with shells. Eggs that didn't need to be deposited in water. Eggs that carried their own little aquatic environment inside them, sealed away from the dry, hostile air. These were the amniotes, and their innovation freed vertebrates from their dependence on water for reproduction. And among those early amniotes, somewhere around 312 million years ago, a split occurred. One lineage would eventually lead to the reptiles and birds. The other would lead to the mammals. And each lineage would take the vertebrate brain in a remarkably different direction, solving similar problems with different neural architectures, like 2 engineering firms given the same design brief, but coming up with completely different solutions. The reptilian approach favored efficiency, lean, streamlined brains that were very good at the specific things they needed to do. The mammalian approach, well, that's where things start to get really interesting, because the mammalian lineage didn't just build a bigger brain. They built a fundamentally different kind of brain. And the story of how that happened is one of the most fascinating engineering tales in the history of life. So let's go back to those early proto-mammals, the synapsids. Now, the word synapsid doesn't get a lot of love in popular culture. You hear about dinosaurs all the time. But synapsids, they actually ruled the Earth first. Before the dinosaurs ever showed up, synapsids were the dominant large land animals for tens of millions of years. All through the Permian period, roughly 300 to 250 million years ago. Some of them were massive. Dimetrodon, that famous animal with the big sail on its back, the one that always gets put in dinosaur toy sets even though it's not a dinosaur at all, that was a synapsid. And here's the thing that might surprise you. Dimetrodon is more closely related to you than it is to any dinosaur. It's on your branch of the family tree, not theirs. Now, the brains of these early synapsids were still fairly reptilian in their basic layout. They had the standard vertebrate toolkit, hindbrain, midbrain, forebrain. And the forebrain was still relatively modest, but something was already starting to shift. The pallium, that sheet of tissue on the roof of the forebrain that we talked about earlier, the one that started out processing smell. In the synapsid lineage, it was beginning to expand, not dramatically at first. Just a little thicker here, a few more neurons there, but the trajectory was set. And then came the worst day in the history of life on Earth, the Permian-Triassic extinction. About 252 million years ago, this was the big one. Not the asteroid that killed the dinosaurs. That was bad. But this was worse. The Great Dying, they call it. Somewhere around 90 to 96% of all marine species went extinct. On land, it was devastating too. Maybe 70% of tetrahedral vertebrate species gone. The leading theory involves massive volcanic eruptions in what's now Siberia. The Siberian traps pouring out lava and greenhouse gases on a scale that's hard to comprehend. The climate swung wildly. Oceans acidified. Oxygen levels may have dropped. It was, by any measure, the closest life on Earth has come to being completely wiped out. And the synapsids took a terrible hit. The big ones, the dominant ones, were largely swept away. But some survived. And the survivors tended to be small. Think about what that means for a moment. When you're a small animal in a devastated world, you face a very specific set of challenges. You lose heat fast because of your surface area to volume ratio. You need to eat frequently because your metabolism runs hot relative to your body size. You're vulnerable to predators. And so the selective pressures on these small surviving synapsids, the ones that would eventually become the cynodonts, and then the true mammals. Those pressures started pushing brain evolution in some very particular directions. First, there was the sensory revolution. These little creatures were probably nocturnal, or at least crepuscular, active at dawn and dusk. And the reason is pretty straightforward. The dinosaurs were taking over the daytime. After the Permian extinction, the Archosaur lineage, the one leading to dinosaurs, recovered faster and came to dominate the large-bodied daytime ecological niches. The early mammals were pushed into the margins. The night shift, essentially. And if you're a small, warm-blooded animal scurrying around in the dark, what senses matter most to you? Not vision, at least not color vision. You need hearing, you need smell, and you need touch. The ability to feel your way through a dark, complex environment. Here's where something truly remarkable happened in the mammalian skull. And it tells us a lot about how brains evolve. In reptiles, the jaw is made up of several bones. In mammals, the jaw is a single bone, the dentary. So where did the other bones go? They shrank over millions of years through a beautifully documented fossil sequence. And this is one of the best documented transitions in all of paleontology. Those extra jaw bones migrated and miniaturized until they became the tiny bones of the mammalian middle ear. The malleus and the incus, two of the three smallest bones in your body right now, used to be jaw bones in your ancestors. Think about that. Bones that once helped your distant relatives bite and chew were repurposed over geological time into an exquisitely sensitive sound amplification system. And this wasn't just a structural change. It was a brain change. Because once you have this incredible new hearing apparatus, you need neural machinery to process all that acoustic information. The auditory cortex expanded. New processing circuits developed. The mammalian brain was being reshaped by the demands of the nocturnal world. And hearing was a huge part of that. Smell was the other big one. The olfactory system in early mammals became extraordinarily sophisticated. And remember, the forebrain's pallium, the tissue that would eventually become the cerebral cortex, had originally evolved to process olfactory information. So when smell became even more critical for survival, the pallium expanded further. More layers of neurons, more complex circuitry. The neocortex, that six-layered sheet of tissue that is the crowning achievement of mammalian brain evolution, almost certainly began its expansion in service of smell. It's a humbling thought, honestly. The part of your brain that lets you do calculus and compose symphonies and contemplate the nature of consciousness. It got its start because your ancestors needed to be really, really good at sniffing things out in the dark. Now, let's talk about that six-layered structure for a second, because it's genuinely one of the most elegant pieces of biological engineering you'll ever encounter. The neocortex. Neo, meaning new, cortex, meaning bark or rind, because it's the outer layer, is organized into 6 distinct horizontal layers, each with different types of neurons and different connectivity patterns. Layer 4, for instance, is where most of the incoming sensory information arrives. Layers 2 and 3 are heavily involved in communication between different cortical areas. Layer 5 sends major outputs down to subcortical structures and the spinal cord. Layer 6 projects back to the thalamus, which is the brain's great relay station. And here's what's really clever about this design. The same basic 6-layer circuit is repeated across the entire neocortex. Whether you're looking at the part that processes vision, or hearing, or touch, or language, or planning, the same fundamental columnar architecture, copied millions of times, handling completely different kinds of information. It's like discovering that a single, versatile circuit board design can be used for everything from audio processing to video rendering to running complex calculations, just by changing the inputs and outputs and tweaking some of the connection weights. Vernon Mountcastle, the Johns Hopkins neuroscientist, proposed this idea of the cortical column as a repeating computational unit back in the 1950s and 60s. And while the details have been debated and refined in the decades since, the core insight holds up. The neocortex is remarkably uniform in its basic structure, and that uniformity is a feature, not a bug. Why? Because it means the neocortex is scalable. If a particular function needs more processing power, say, an animal relies heavily on its whiskers and needs extremely detailed tactile maps, evolution can simply expand the relevant cortical area without having to invent a whole new type of neural tissue. Just add more columns. It's modular, it's extensible, and that modular design is a huge part of why the mammalian brain was able to grow and diversify so dramatically over the next 200 million years. But we're getting ahead of ourselves. Back in the Mesozoic, during the age of dinosaurs, mammals were still mostly small. For over 100 million years, They lived in the shadows of the great reptiles. And yet, during all that time, their brains were quietly evolving. Fossil endocasts, those natural casts of the inside of the skull that preserve the shape of the brain, show us that even these tiny Mesozoic mammals had relatively large brains for their body size compared to the reptiles around them. A study published in Science in 2011 by Tim Rowe and colleagues at the University of Texas used CT scanning to examine the skulls of some of the earliest known mammals like Morganucodon and Hadrocodium, and they found that brain expansion happened in 3 major pulses. The first pulse was driven by improvements in olfaction, a bigger olfactory bulb, more olfactory cortex. The second was driven by touch, particularly the processing of information from body hair, which was a new mammalian innovation. Whiskers, fur. These are sensory organs, and they feed into the somatosensory cortex. The third pulse involved neuromuscular coordination and motor control. So the mammalian brain wasn't just getting bigger randomly. It was getting bigger in response to specific ecological pressures, each expansion tied to a concrete survival advantage. Smell the predator before it sees you. Feel the vibrations in the ground through your whiskers. Coordinate your muscles for quick, agile escape through the underbrush at night. Every bit of neural expansion paid for itself in survival. And this is an important principle in brain evolution that's worth sitting with for a moment. Brains are expensive, incredibly expensive. Your brain right now accounts for about 2% of your body weight, but consumes roughly 20% of your resting metabolic energy. That's an enormous cost. And in the ruthless economy of natural selection, you don't maintain expensive tissue unless it's earning its keep. So every time we see brain expansion in the fossil record, we should ask, what was the payoff? What survival advantage justified that metabolic investment? Because there had to be one. Evolution doesn't do charity. Now, while the mammals were quietly refining their neural hardware in the Mesozoic shadows, the other lineage, the reptiles and their descendants, was doing something interesting too. And I want to take a brief detour here because it helps us appreciate the mammalian brain. By contrast, birds which evolved from theropod dinosaurs developed their own form of intelligence, and they did it without a neocortex. Bird brains are organized very differently. Instead of the layered cortical sheets that mammals have, birds have clusters of neurons organized into nuclei, dense, ball-like structures. For a long time, scientists assumed this meant bird brains were primitive, that birds were basically running on instinct. The old term for the bird forebrain was neostriatum, implying it was equivalent to the relatively simple striatum in mammals. But that turned out to be spectacularly wrong. In 2005, a consortium of neuroscientists formally renamed most of the structures in the bird brain to reflect the growing understanding that many of these regions are actually equivalent to mammalian cortical areas. They're just organized differently. And when you look at what birds can actually do, crows making and using tools, parrots understanding abstract concepts, scrub jays planning for the future, it's clear that the avian brain is a powerful cognitive organ. It just solved the engineering problem differently. Nuclei instead of layers. clusters instead of sheets. Different architecture, comparable results in many domains. This is one of those beautiful lessons that keeps coming up in engineering. Whether we're talking about biological systems or human-designed ones, there's almost never only one way to solve a problem. The mammalian neocortex and the avian pallium are like two completely different computer architectures that can both run sophisticated software, and understanding that helps us appreciate that the mammalian approach, the layered, column-tar scalable neocortex, isn't the only path to complex cognition. It's one path, but it happens to be our path, and it's the one that eventually led to the most dramatically enlarged brain the world has ever seen. Which brings us to the event that changed everything for mammals. 66 million years ago, an asteroid roughly 10 to 12 kilometers across slammed into what is now the Yucatan Peninsula in Mexico. And the world changed in ways that are almost impossible to exaggerate. The Chicxulub impactor, that's what we call it now, named after the town near the center of the crater, struck with an energy roughly equivalent to 10 billion Hiroshima bombs. 10 billion. The rock itself was traveling at something like 20 kilometers per second. And when it hit the shallow seas of the Yucatan, it punched a crater nearly 180 kilometers across and sent a column of superheated debris rocketing back out through the atmosphere and into space. That debris rained back down across the entire planet, heating the upper atmosphere to the point where forests ignited across whole continents. Tsunamis hundreds of meters tall radiated outward from the impact site. Sulfur-rich rocks at the impact location vaporized and filled the atmosphere with aerosols that blocked sunlight for months, maybe years. Global temperatures plummeted, photosynthesis collapsed, and roughly 75% of all species on Earth went extinct, including every non-avian dinosaur. Now, think about what that means for the story we've been following. For over 100 million years, mammals had been living in the shadows, literally. Small, nocturnal, hiding in burrows and underbrush, eating insects, staying out of the way of creatures that would crush them without noticing. Those survival pressures, as we talked about, had shaped their brains in specific ways. Better hearing, better smell, a growing neocortex that could process complex sensory information in the dark. But the ecological niches available to them were limited. You can't grow very large when there's a 2-ton theropod patrolling your neighborhood. You can't become a daytime predator when the daytime already belongs to something faster and bigger than you. The dinosaurs were a ceiling, and mammals were pressed flat against the floor beneath it. And then, in a geological instant, the ceiling was gone. What happened next is one of the most extraordinary radiations in the history of life. Within the first few 100,000 years after the impact, which sounds like a long time but is genuinely a blink in evolutionary terms, mammals began diversifying at a pace that the fossil record rarely shows for any group. They moved into niches that had been occupied by dinosaurs for 160 million years. Some got big, some got really big, some took to the trees, some took to the water, some took to the air, and their brains changed with them, because every new niche presented new cognitive demands. Here's what's really fascinating about this period. which paleontologists call the Paleocene. The roughly 10 million years right after the extinction. For a while, the mammals that dominated weren't the ones you'd expect. The early Paleocene was ruled by groups that are mostly extinct now. Condylarths, which were generalized herbivores and omnivores, and creodonts, which were early predatory mammals. Their brains, based on endocysts, those natural casts of the inside of the skull that preserve the brain's shape, were relatively small compared to their body size. The ratio of brain to body, what neuroscientists call the encephalization quotient, was actually pretty modest in these early post-dinosaur mammals. They'd inherited the basic mammalian brain plan, with its expanding neocortex, but they hadn't yet been pushed to develop it further. In a world emptied of competitors, you didn't necessarily need to be all that clever. There was food everywhere, and not much competition for it. But that didn't last. As ecosystems recovered and became more complex, as more species filled more niches and began competing with each other, the pressure on brains ramped up again. And this is where you start to see the lineages that would eventually lead to modern mammals really come into their own. The primates, the carnivorans, the cetaceans, the rodents. Each group found its own way to expand and elaborate the neocortex, and each did it in response to the specific demands of its ecological niche. Let's follow the primate line, because that's our line. And it's where the brain story gets personal. The earliest primates, or at least the earliest animals that we can confidently place near the primate lineage, appear in the fossil record right around the time of the extinction, maybe just before it. There's a tiny creature called Purgatorasius, known from teeth and partial jaw fragments found in Montana, dating to within a few 100,000 years of the KPG boundary. It was small, probably weighed about 40 grams, roughly the size of a mouse, and it was likely an arboreal insectivore, living in trees, eating bugs and maybe fruit. And here's the thing about living in trees. It changes everything about what your brain needs to do. Think about the computational demands of moving through a three-dimensional environment of branches. You need depth perception, really good depth perception. Because if you misjudge a jump by even a few centimeters, you fall, and falling from a tree canopy is often fatal. You need precise hand-eye coordination because you're gripping branches of varying thickness and texture. You need to be able to judge distances, track moving targets like insects, and make split-second decisions about which branch will hold your weight. The arboreal niche is, in a very real sense, a cognitive gymnasium. Matt Cartmill, a biological anthropologist at Boston University, proposed what he called the visual predation hypothesis back in the 1970s. His idea was that the defining features of early primate evolution, forward-facing eyes giving stereoscopic vision, grasping hands with nails instead of claws, and enhanced visual processing in the brain, evolved together as adaptations for catching insects on thin branches in the forest canopy. It's a beautifully integrated argument. You need the eyes to judge the distance to the insect. You need the hands to grab it while holding onto a branch. And you need the brain to coordinate all of that in real time. Now, not everyone agrees with Cartmill's hypothesis in its original form. Robert Sussman proposed an alternative the angiosperm coevolution hypothesis, suggesting that primates evolved their traits in response to the spread of flowering plants and the fruits and nectar they produced. And there are other ideas too. But what nearly everyone agrees on is that the arboreal environment was a major driver of primate brain evolution, particularly the expansion of visual processing areas in the neocortex. And you can see this in the fossils. By the Eocene, roughly 56 to 34 million years ago, you get primates that are unmistakably primates. Creatures like adipids and omomiids. These are the two big groups of early true primates, and their skulls show something remarkable. Compared to other mammals of similar body size, their brains are noticeably larger, and the expansion is concentrated in the visual cortex and the frontal areas. The olfactory bulbs, which had been so important to those early nocturnal mammals, are actually getting relatively smaller. The brain is being reorganized. Vision is taking over from smell as the dominant sense, and the neocortex is expanding to handle all that visual information. This is a pattern you see again and again in brain evolution. It's not just about getting bigger, it's about rebalancing. Resources shift from one sensory system to another as the animal's lifestyle changes. The early mammals invested heavily in smell and hearing because they were nocturnal. The early primates began investing in vision because they were arboreal and increasingly diurnal, active during the day. And every time you expand one area of the cortex, you create opportunities for new connections, new circuits, new cognitive capabilities that might not have anything directly to do with the original sensory function. It's like adding more rooms to a house and then discovering that the extra space lets you do things you never planned for. By the late Eocene and into the Oligocene, around 30 to 25 million years ago, the primate lineage had split into two major groups. You had the strepsirrhines, the lemurs, lorises, and their relatives, and the haplorhines, which includes tarsiers, monkeys, apes, and eventually us. The haplorhine line is the one where brain expansion really accelerated, and a big part of the reason has to do with something that might surprise you. It's not just about navigating trees or finding food. It's about other primates. This is where we get into what's sometimes called the social brain hypothesis, and it was most influentially articulated by Robin Dunbar, an anthropologist and evolutionary psychologist at Oxford. Dunbar noticed something striking in the 1990s when he plotted neocortex size against social group size across different primate species. There was a remarkably tight correlation. The larger the typical social group for a species, the larger its neocortex relative to the rest of the brain. Not the whole brain, specifically the neocortex, the part that handles complex information processing, planning, and abstract thought. And when you think about it, this makes deep intuitive sense. Living in a social group is cognitively demanding in ways that are completely different from navigating a physical environment. You have to keep track of relationships, not just your own relationships with other individuals, but the relationships between other individuals. Who's allied with whom? Who's feuding? Who can be trusted? Who's likely to cheat? Who's dominant? Who's submissive? And how those hierarchies shift over time. You have to remember past interactions, predict future behavior, and adjust your own behavior strategically. It's like playing a never-ending game of chess where the pieces have their own agendas and the rules keep changing. Dunbar estimated that for humans, the correlation predicts a natural social group size of about 150, a number that's now famous as Dunbar's number, and you see it echoed in all sorts of human social structures. From the size of hunter-gatherer bands to military companies to the number of people, most of us can maintain meaningful relationships with, whether or not you buy the exact number. The broader point is compelling. Social complexity may have been one of the most powerful engines driving the expansion of the primate neocortex. And this is where the story starts to narrow toward our own lineage. Because something happened in Africa somewhere around 6 to 7 million years ago that set one particular group of apes on a trajectory unlike anything evolution had produced before. The common ancestor of humans and chimpanzees, and we still don't have a definitive fossil for this ancestor, though candidates like Sahelanthropus chadensis, discovered by Michel Brunet's team in Chad in 2001, are tantalizing. This ancestor's descendants split into at least 2 lineages. One led to the chimps and bonobos, the other led to us. And on our side of that split, brains began to grow in a way that was, frankly, unprecedented in the entire history of life on this planet. But the growth didn't happen all at once, and the reasons behind it are still fiercely debated. And the story of how we went from a brain, roughly the size of a chimpanzee's, to the 3-pound organ sitting inside your skull right now. That story involves climate change, walking upright, tools made of stone, the control of fire, and a feedback loop so powerful that it essentially rewired the relationship between biology and behavior. So, let's talk about that feedback loop, because it's one of the most remarkable things in all of evolution. And once you see it, you start to understand why our lineage, and really only our lineage, ended up with brains this absurdly large. Picture the situation. You're an ape living in East Africa somewhere around 6 million years ago. The climate is shifting. Forests are thinning. Grasslands are expanding. The lush, continuous canopy your ancestors swung through is breaking apart into patches. And between those patches there's open ground. Savanna, scrubland, places where a tree-dwelling ape is exposed and vulnerable. Now there's a long-standing idea, going back to Raymond Dart in the 1920s, that bipedalism, walking upright on 2 legs, evolved as a response to this kind of environmental change. The logic is intuitive. If you need to cross open ground between forest patches, being able to stand up and walk efficiently is a real advantage. You can see over tall grass. You can carry things with your hands. And critically, walking on 2 legs is far more energy efficient than knuckle walking when it comes to covering long distances. A study by Michael Sokol and David Reichlin, published in 2007, actually measured this directly. They put chimps on treadmills, which is exactly as entertaining as it sounds, and confirmed that bipedal walking in humans uses about 75% less energy than quadrupedal walking in chimps, covering the same distance. But here's the thing. Bipedalism didn't just change how we moved. It changed what we could do with our hands. And what we could do with our hands changed what we could do with our brains. And what we could do with our brains changed what we could do with our hands. You see where this is going? It's circular. It feeds on itself. And that self-reinforcing cycle between locomotion, manual dexterity, tool use, and brain expansion is the engine that drove hominin evolution for millions of years. The earliest hominins we have good fossil evidence for, creatures like Ardipithecus ramidus, described by Tim White's team from Ethiopian sites dating to about 4.4 million years ago, were already showings and signs of bipedal adaptation, though Ardi, as the most complete specimen is nicknamed, was probably still spending a lot of time in trees. Her brain was small, roughly 300 to 350 cubic centimeters, which is right in the range of a modern chimpanzee. So bipedalism came first, the brain expansion came later. That sequence matters because it tells you that walking upright wasn't a consequence of getting smarter. It was one of the preconditions for it. Then you get the Australopithecines. And this is where things start to get really interesting. Australopithecus afarensis, Lucy's species, famously discovered by Donald Johanson and Tom Gray in the Afar region of Ethiopia in 1974, lived between about 3.9 and 2.9 million years ago. Lucy's brain was around 400 to 450 cubic centimeters. Still small, still chimp-sized, more or less. But her skeleton tells you she was a committed biped. She walked upright habitually. And the Latoli footprints in Tanzania, preserved in volcanic ash about 3.6 million years ago, discovered by Mary Leakey's team, confirm it. Those footprints look remarkably like yours or mine. No divergent big toe, a clear arch, a heel strike followed by a toe off. These were creatures walking like humans, even if they didn't yet think like humans. So for roughly the first 2 to 3 million years of our lineage, brains stayed relatively modest. Bipedalism was established, but the cognitive explosion hadn't started yet. What changed? Well, around 2.6 million years ago, something new appears in the archaeological record. Stone tools. The earliest well-dated stone tools come from a site called Gona in Ethiopia, and they're simple but deliberate. Sharp flakes struck from cobbles, what archaeologists call the Oldowan industry, named after Olduvai Gorge in Tanzania, where Louis and Mary Leakey found similar tools in the 1960s. These aren't accidental breaks. When you examine them, you can see that whoever made them understood how stone fractures. They chose the right kind of rock, usually basalt or chert, and struck it at the right angle to produce a sharp edge. That requires planning. It requires an understanding of cause and effect, and it requires fine motor control in the hands and fingers that's governed by expanded regions of the motor cortex and the cerebellum. And here's where the feedback loop really kicks in. Making tools requires brain power, but having tools, especially sharp cutting tools, gives you access to food resources you couldn't exploit before. Meat, specifically. With a sharp stone flake, you can butcher a carcass. You can cut through hide that your teeth can't penetrate. You can crack bones for marrow, which is one of the most calorie-dense and nutrient-rich foods available on the African savanna. And more calories especially more animal fat and protein, can support a larger, more metabolically expensive brain. Remember what we talked about earlier. The brain is only about 2% of your body weight, but it consumes roughly 20% of your resting energy. Growing a bigger brain is metabolically costly. You need a reliable source of dense calories to fuel it. Meat and marrow provided exactly that. Leslie Aiello and Peter Wheeler proposed this connection formally in 1995 with what they called the expensive tissue hypothesis. Their insight was elegant. The brain isn't the only metabolically expensive organ in the body. The gut is too. And if you shift to a higher quality diet, more meat, more fat, more nutrient-dense food, you can afford to have a smaller gut, because you don't need the enormous digestive tract that a folivore or a frugivore requires to extract nutrients from low-quality plant food, and the metabolic savings from shrinking the gut can be redirected to growing a bigger brain. It's a trade-off. Gut size goes down, brain size goes up. Total metabolic budget stays roughly balanced. And when you look at the fossil record, that's broadly what you see. As hominin brains expanded, their teeth got smaller, their faces got flatter, their guts apparently shrank. The anatomy tells the same story the theory predicts. Now, the species most associated with this transition is Homo habilis, handyman, named by Louis Leakey, Philip Tobias, and John Napier in 1964. Habilis lived roughly 2.4 to 1.4 million years ago, and its brain was noticeably larger than the Australopithecines, around 500 to 700 cubic centimeters. That's a significant jump, and habilis is found in association with those Oldowan stone tools, though there's ongoing debate about whether Australopithecines might have been making tools too. A 2015 paper reported cut marks on bones from Dikika, Ethiopia, dating to 3.4 million years ago, which would push tool use back into Lucy's time. The evidence is contested, but it's a reminder that these transitions were probably gradual and messy, not clean and sudden. But the real star of the next act is Homo erectus. And this is a species that deserves some time, because erectus is arguably the most successful hominin that ever lived, besides us. And honestly, the jury's still out on that comparison, given how long erectus lasted, appearing in the fossil record around 1.9 million years ago. In Africa, Homo erectus had a brain ranging from about 600 to over 1,000 cubic centimeters. A dramatic increase. Erectus was tall, relatively long-legged, with body proportions much closer to modern humans than anything that came before. This was a creature built for endurance, walking and running across open landscapes. And erectus did something no hominin had done before. It left Africa. Fossils of Homo erectus have been found in Georgia. The Dmanisi skulls, dating to about 1.8 million years ago, discovered in the 1990s. In Java, in China, across a vast geographic range, erectus spread across the Old World and persisted for well over 1 million years, possibly until as recently as 100,000 years ago in Southeast Asia. That's an extraordinary run. And during that time, erectus developed more sophisticated stone tools. The Acheulean hand axe, that beautiful teardrop-shaped tool that's been called the Swiss Army knife of the Pleistocene. Making an Acheulean hand axe is genuinely difficult. You have to hold a mental template of the finished shape in your mind and remove flakes systematically from both sides of a stone to achieve bilateral symmetry. Neuroscientists like Dietrich Stout have used brain imaging to study what happens when modern humans learn to make these tools, and the results are fascinating. It activates regions involved in planning, spatial reasoning, and motor sequencing, including parts of the prefrontal cortex and Broca's area, which in modern humans is associated with language, and that connection between tool-making and language is one of the most provocative ideas in paleoanthropology. The neural circuits you need to plan and execute a complex sequence of hand movements overlap significantly with the circuits involved in producing and comprehending structured sequences of sounds, which is essentially what language is. So, the same selective pressures that rewarded better tool-making may have simultaneously been laying the neural groundwork for speech. Not language as we know it, not yet, but the cognitive infrastructure that language would eventually run on. And then there's fire. This is another one of those game changers that's hard to overstate. The earliest convincing evidence for controlled use of fire comes from a site called Wonderwork Cave in South Africa, dating to about 1 million years ago, with some researchers pushing the evidence even earlier. Richard Wrangham, a primatologist at Harvard, built an entire theory around fire and cooking. His cooking hypothesis, laid out in his 2009 book, Catching Fire. Wrangham's argument is that cooking food was a pivotal adaptation because it dramatically increases the caloric yield of both meat and plant foods. Cooking breaks down tough fibers, denatures proteins, gelatinizes starches. It essentially pre-digests food before it enters your body. That means you extract more energy from less food, and you spend less energy on digestion, which means, once again, more metabolic budget available for the brain. Think about what that means in practical terms. A raw potato gives you significantly fewer usable calories than a cooked one. Raw meat takes more chewing and more digestive effort than cooked meat. Cooking is, in a very real sense, an external digestive technology. And if Wrangham is right, then fire didn't just keep our ancestors warm and safe from predators at night. It literally fueled the expansion of the brain. It's technology reshaping biology. The tool changes the toolmaker. And all of this, bipedalism freeing the hands, hands making tools, tools accessing better food supporting bigger brains, fire unlocking and even more calories, and all of it is happening against a backdrop of intense social complexity. These hominins lived in groups. They cooperated. They competed. They needed to track alliances, read intentions, anticipate what others would do. Dunbar's social brain hypothesis, which we touched on before, suggests that managing all of this social information was itself a powerful driver of brain expansion. So you've got multiple pressures all pushing in the same direction, all reinforcing each other. And the result is a trajectory of brain growth that, on the timescale of evolution, looks almost vertical. By the time you get to the later stages of the genus Homo, species like Homo heidelbergensis, appearing around 700,000 years ago, with brain volumes pushing 1,200 cubic centimeters, and beyond. You're looking at creatures that are almost, but not quite, us. They hunted cooperatively. They may have used wooden spears. The Schöningen spears from Germany, dating to about 300,000 years ago, are beautifully crafted throwing weapons, balanced and aerodynamic. These weren't clumsy proto-humans stumbling around. They were sophisticated, coordinated hunters with complex social lives and brains approaching modern human size. And somewhere in this population, or one closely related to it, the lineage that would become Homo sapiens was taking shape. The oldest fossils assigned to our species come from a site that, when it was announced in 2017, surprised almost everyone, because it wasn't in East Africa where so many hominin fossils had been found. It was in Morocco, at a place called Jebel Irhoud, a cave site in the hills west of Marrakesh, where a team led by Jean-Jacques Hublin had been working for years, carefully redating and reanalyzing fossils that had actually been found decades earlier, but whose significance hadn't been fully appreciated. What Hublin's team showed was that these fossils, skulls, mandibles, stone tools, were approximately 300,000 years old, pushing the origin of Homo sapiens back by 100,000 years from what most textbooks had been saying. And the location mattered just as much as the date. Morocco is in North Africa, about 4,000 kilometers from the East African Rift Valley sites that had dominated the story of human origins for so long. The implication was profound. Our species didn't emerge from a single Garden of Eden somewhere in the Ethiopian highlands. Instead, the picture that's been forming, and Jebel Irhoud was a key piece of this, is that Homo sapiens evolved across the African continent in multiple populations that were connected but geographically dispersed, exchanging genes and ideas across vast distances over tens of thousands of years. Some researchers call this the African multi-regionalism model, or the Pan-African origin hypothesis, and it's a much messier, more interesting story than the old simple narrative. Now, here's what's really fascinating about the Jebel Irhoud skulls. If you look at the faces, they're remarkably modern. The brow ridges are reduced compared to Homo heidelbergensis. The midface is flat, tucked under the brain case, rather than projecting forward. You'd recognize it as a human face. But the brain case itself, the part that houses the brain, is elongated, more like a football shape than the round globe you and I carry around. Modern human skulls are distinctively globular. We have this high, rounded vault with expanded parietal bones on the sides and a bulging forehead up front. The Jebel Irhoud people didn't have that yet. Their brains were probably close to modern size, around 1,300 cubic centimeters or so, but the shape was different, and that distinction between size and shape turns out to be enormously important. A team led by Simon Neubauer working with Hublin published a study in 2018 using CT scans of fossil skulls and computational reconstructions to track how brain shape changed over the course of Homo sapiens evolution. What they found was that the globular brain case we have today didn't appear all at once. It evolved gradually, reaching its modern form only around 100,000 to 35,000 years ago. Think about what that means. The earliest members of our species had brains that were roughly the same size as ours, but organized differently, shaped differently, and presumably wired differently in at least some respects. The brain was still being sculpted, still being refined, tens of thousands of years after Homo sapiens first appeared in the fossil record. So what was changing inside that reshaping skull? This is where it gets speculative, but in the most interesting way. The globularization of the brain case seems to involve expansion of the parietal regions and the cerebellum. The parietal cortex is involved in spatial reasoning, attention, working memory, integrating information from different senses. The cerebellum, which we talked about way back when we were discussing fish, has turned out to be far more than a motor coordination center. It's deeply involved in language processing, planning, and cognitive flexibility. So the suggestion is that, even after our species existed, the brain was still undergoing significant developmental changes that may have affected cognition in important ways. And this brings us to one of the biggest questions in all of human evolution, one that people argue about passionately. What happened around 70 to 50,000 years ago? Because something seems to shift. In the archaeological record, you start seeing an explosion of what archaeologists call behavioral modernity. Complex symbolic artifacts, jewelry, shell beads with holes drilled through them, strung on cords and worn around the neck, ochre pigments ground up and apparently used for body decoration, or maybe for art, bone tools finely made, fishing hooks, evidence of long-distance trade networks, with materials showing up hundreds of kilometers from their geological source, and then, of course, cave art. The stunning paintings at places like Chauvet in France, dating to around 36,000 years ago, or the even older hand stencils and animal figures found in caves on the island of Sulawesi in Indonesia, some of which may be over 45,000 years old. For a long time, this was called the Upper Paleolithic Revolution, or the Great Leap Forward, a phrase popularized by Jared Diamond, and the idea was that something happened, some kind of neurological mutation or rewiring event, that suddenly gave Homo sapiens the capacity for fully modern, symbolic thought. One day we were cognitively archaic, the next we were painting masterpieces on cave walls. But that story has gotten a lot more complicated. Finds from Africa, particularly from sites like Blombos Cave in South Africa, have pushed evidence of symbolic behavior much further back. The Blombos ochre pieces, with their deliberately engraved geometric patterns, date to around 77,000 years ago. Shell beads from North Africa are even older, perhaps 100,000 years or more. So it's not that there was a single dramatic moment. It's more like these capacities were developing gradually, appearing in different populations at different times, sometimes flickering into visibility in the archaeological record and then disappearing. Maybe because populations were small and vulnerable, and cultural innovations could be lost when groups died out or fragmented. This is a really important idea, and it connects to something you might not immediately think of as relevant to brain evolution: demography, population size. The argument made compellingly by researchers like Adam Powell, Stephen Shanahan, and Mark Thomas is that the accumulation and maintenance of complex culture depends not just on having smart enough brains, but on having enough people in contact with each other to sustain the transmission of knowledge. A small, isolated group might invent something brilliant, but if there aren't enough people to learn it, practice, teach it to the next generation, and improve on it, that innovation can vanish. It's like a fire that needs a certain density of fuel to keep burning. So, the apparent acceleration of cultural complexity around 50,000 years ago might not reflect a sudden change in brain hardware at all. It might reflect a demographic threshold. Populations growing large enough and connected enough to sustain cumulative culture. But let's not get too far ahead of ourselves, because there's another character in this story who deserves real attention, and that's the Neanderthals. We've been focused on the Homo sapiens lineage, but for hundreds of thousands of years, Europe and Western Asia were home to these remarkable people, Homo neanderthalensis, who had brains that were, on average, slightly larger than ours. Let that sink in. Neanderthal brain volume averaged around 1,400 to 1,500 cubic centimeters, compared to about 1,350 for modern humans. They were not the dim-witted brutes of old cartoons. They made sophisticated stone tools. The Mousterian tradition, with its carefully prepared Levallois cores, they controlled fire. They buried their dead at least sometimes. There's evidence they used medicinal plants, that they made adhesives from birch bark pitch to haft stone points onto wooden shafts, a process that requires careful temperature control and multiple steps. They survived ice ages in some of the harshest environments on Earth for over 300,000 years, and yet their brains were shaped differently from ours. Remember that globularization we talked about? Neanderthal brain cases were long and low, with prominent brow ridges and a distinctive occipital bun, a bulge at the back of the skull. Their brains were large, but the proportions were different. They appear to have had relatively larger occipital lobes, the visual processing areas at the back, and possibly smaller parietal and temporal regions compared to modern humans. Eilund Pierce and Robin Dunbar published a study in 2013 suggesting that Neanderthals may have devoted more neural real estate to vision and body control, possibly as an adaptation to the lower light levels of high-latitude European environments, and correspondingly less to the social cognition networks that seem to be expanded in Homo sapiens. It's a hypothesis, not a settled fact, but it's a thought-provoking one. Two different solutions to the problem of being a large-brained hominin, two different architectures. And here's where the story takes one of its most dramatic turns. Because these two lineages met when Homo sapiens expanded out of Africa, and we now know there were multiple waves of migration, not just one. They encountered Neanderthals in the Middle East, in Europe, in Central Asia, and they didn't just compete with them. They interbred. The evidence for this came from ancient DNA, and it was one of the great scientific revelations of the early 21st century. Svante Pääbo, a Swedish geneticist working at the Max Planck Institute in Leipzig, had spent decades developing techniques to extract and sequence DNA from ancient bones, a task that most people thought was impossible, because DNA degrades over time breaks into tiny fragments, and gets contaminated with microbial DNA. The technical challenges were staggering. But Paabo and his team, through years of painstaking methodological refinement, managed to sequence the Neanderthal genome, publishing a draft in 2010. And when they compared it to modern human genomes, they found something that shocked the field. People of non-African descent carry roughly 1 to 4% Neanderthal DNA. You, if your ancestry traces through Europe or Asia, are carrying genes that came from Neanderthals. Genes that entered the Homo sapiens gene pool through interbreeding events that probably occurred somewhere in the Middle East around 50 to 60,000 years ago. Paabo won the Nobel Prize in Physiology or Medicine in 2022 for this work, and he deserved it. But the implications go far beyond just knowing that interbreeding happened. Some of those Neanderthal gene variants appear to have been beneficial and were positively selected for in human populations. Variants affecting immune function, skin and hair characteristics, adaptation to different climates, and some may have affected the brain. There are Neanderthal-derived gene variants that has been associated with skull shape, with pain sensitivity, with sleep patterns. The picture is still being filled in, but the point is that our genome is not purely sapiens. It's a mosaic, and the same is true for another group, the Denisovans, known mostly from a single finger bone and a few teeth found in a cave in Siberia, whose genome Paabo's team also sequenced, and whose DNA shows up in modern populations in Southeast Asia, Melanesia, and Australia, sometimes at even higher percentages than Neanderthal DNA. So the modern human brain, your brain, the one you're using right now to process these words, or maybe to drift gently toward sleep, is the product of not just one lineage's evolutionary journey, but a confluence of lineages: sapiens, Neanderthal, Denisovan, and possibly others we haven't identified yet. It's a brain shaped by 300,000 years of sapiens evolution, but carrying echoes of far older stories, older architectures, older solutions to the problem of surviving in a complicated world. And what that brain can do, what it started doing with increasing intensity over the last 100,000 years, is something genuinely without precedent in the history of life on Earth. Because at some point, biological evolution stopped being the main driver of change in how humans interact with their environment. Cultural evolution took over, and cultural evolution operates on a completely different timescale. Genetic evolution works across generations, thousands, tens of thousands, hundreds of thousands of years for significant changes to accumulate. Cultural evolution can work within a single lifetime. An idea can spread across a continent in a generation. A technology can transform the conditions of existence in a few decades. And the engine of all of this, the thing that makes cumulative culture possible, is language. That's the engine. Language is what makes cumulative culture possible. And it's worth sitting with that idea for a moment, because language isn't just communication. Lots of animals communicate. Vervet monkeys have distinct alarm calls for different predators. One call for eagle, another for leopard, another for snake. Honeybees perform waggle dances that encode the direction and distance of food sources with remarkable precision. Dolphins have signature whistles that function almost like names. But none of that is language in the way you and I use the word. What human language does, and what no other communication system on Earth does with anything close to the same power, is combine a finite set of elements into an infinite number of novel meanings. You can say a sentence that has never been spoken before in the history of the species, and someone hearing it for the first time will understand you. That's not just impressive. That's world-altering, because it means you can describe things that aren't present. You can talk about the past. You can plan for a future that hasn't happened yet. You can explain to someone how to do something they've never seen done. You can tell a story about a person who doesn't exist to illustrate a principle that has no physical form. And that capacity, that ability to transmit complex, structured information from one mind to another, is what turns individual innovation into cumulative culture. Without language, if you figure out a better way to nap a stone tool, maybe your children learn it by watching you. Maybe a few people in your group pick it up through imitation. But the technique is fragile. It can be lost in a generation. With language, you can explain the technique. You can describe why it works. You can tell someone about the mistake you made the first time and how to avoid it. And that means knowledge doesn't just pass from one person to another. It accumulates. Each generation can start from where the last one left off, rather than reinventing everything from scratch. Now, here's the tricky part. We don't know when language evolved, and this is one of the most genuinely frustrating gaps in the whole story of the brain, because language doesn't fossilize. You can't dig up a sentence. You can look for indirect evidence. The shape of the hyoid bone, which supports the larynx and is involved in speech production. There's a Neanderthal hyoid bone from Kebara Cave in Israel, dating to about 60,000 years ago, and it looks remarkably similar to a modern human hyoid, which suggests Neanderthals, at least, had the anatomical equipment for speech. You can look at the FOXP2 gene, which when mutated, causes severe speech and language disorders in humans. Neanderthals had the same version of FOXP2 that we do, which is interesting but doesn't prove they had language. The gene is involved in fine motor control of the mouth and face, and it's found in other species too. You can look at the archaeological record for evidence of symbolic thinking, which many researchers consider a proxy for language. If you can think symbolically, you probably have the cognitive architecture that supports language, and vice versa. Those shell beads from Blombo's cave, the ochre engravings, the cave paintings, they all suggest minds that are manipulating symbols, and it's hard to imagine how you'd have a fully symbolic culture without language to organize and transmit it. But the honest answer is that we don't have a firm date. Some researchers, like Noam Chomsky, have argued that language emerged relatively suddenly, perhaps as a single genetic mutation that rewired the brain in a critical way, what's sometimes called the saltationist view. Others, like Steven Pinker and Ray Jackendorf, have argued for a more gradualist picture, where language evolved incrementally over hundreds of thousands of years, building on earlier proto-linguistic abilities. And there are fascinating intermediate positions. People like Michael Tomasello, who emphasizes that what's really special about human communication isn't just the grammar, but the underlying capacity for shared intentionality, the ability to understand that another person has a mind with its own perspective, and to deliberately share attention and goals with them. That capacity, Tomasello argues, is the foundation on which language was built, and it may have deep roots in our lineage, going back to cooperative behaviors in early Homo. What we can say is that by the time you see the full flowering of behavioral modernity, the explosion of art, technology, long-distance trade networks, elaborate burial practices, language is almost certainly in place, and once it is, the brain doesn't need to change biologically for human behavior to transform. Think about what that means. For the previous 3.5 billion years of life on Earth, if an organism was going to do something new, it needed new biological equipment, a new protein, a new neural circuit, a new body plan. That takes generations. It takes selection pressure and random mutation and vast stretches of time. But a human with language and cumulative culture can learn to do something entirely new in an afternoon, not because their brain has changed, but because someone else figured it out and told them about it. And this is where the story of the brain starts to become the story of civilization. And the two become almost impossible to separate, because the brain you have right now, the one who's settling into the pillow, the one whose electrical rhythms are probably starting to slow just a little, is essentially the same brain that existed in those early Homo sapiens populations 100,000 years ago. The hardware hasn't changed much. What's changed spectacularly is the software, the accumulated cultural knowledge that gets loaded into that hardware from the moment you're born. Let's talk about that for a second, because the way the brain develops is itself one of the most remarkable parts of this story. A human baby is born with roughly the same number of neurons it will have as an adult, about 86 billion, give or take. But the connections between those neurons, the synapses, are far from finished. In the first few years of life, synapses form at an almost unbelievable rate, something like 700 to 1,000 new synaptic connections every second in a toddler's brain. By age 2 or 3, a child has roughly twice as many synapses as an adult. And then something equally important happens: pruning. The connections that get used, that get reinforced by experience, are strengthened and maintained. The ones that don't get used are eliminated. It's a use it or lose it system, and it means that the brain is literally sculpted by experience. The environment, the language you hear, the faces you see, the problems you're asked to solve, the emotional landscape of your early life, physically shapes the wiring of your brain. This is what neuroscientists call experience-dependent plasticity. And it's not unique to humans, but the degree to which human brains depend on it is extraordinary. A kitten's visual cortex needs visual input during a critical period to wire up properly. That's been known since Hubel and Wiesel's Nobel Prize-winning work in the 1960s. But a human brain needs language input, social input, emotional input, and the sheer complexity of what it's wiring up is orders of magnitude greater. This is, in a sense, the evolutionary bargain that our species made. Instead of hardwiring everything Instead of building a brain that comes preloaded with all the behaviors it will ever need, the way an insect's brain largely does. We build a brain that comes preloaded with the ability to learn, to absorb whatever cultural environment it's born into. A baby born in Tokyo and a baby born in Lagos have the same brain. But the cultural software that gets installed, the language, the social norms, the skills, the knowledge, will be profoundly different, and both brains will wire themselves to be optimally suited to their particular world. Here's what's really clever about this arrangement. It means that when the cultural environment changes, the brain can adapt without any genetic evolution at all. When humans invented agriculture, roughly 12,000 years ago in the Fertile Crescent, the brains that had to manage this new way of life were biologically identical to the brains of the hunter-gatherers who came before. But those brains wired themselves differently because the demands were different. You needed to track seasons, plan months ahead, manage stored resources, negotiate land boundaries, coordinate labor among larger groups of people than any hunter-gatherer band had ever sustained. And when writing was invented around 5,000 years ago in Mesopotamia, those little wedge-shaped marks pressed into clay tablets. The brain adapted again. There's no gene for reading. There's no brain region that evolved to process written text. What happens instead, as the neuroscientist Stanislas Dehaene has shown in beautiful detail, is that regions of the visual cortex that originally evolved to recognize objects and faces get repurposed, recycled, in Dehaene's term, to recognize letters and words. The brain's plasticity allows it to do something it was never designed to do, using hardware that evolved for completely different purposes. And this is the pattern that repeats over and over as cultural evolution accelerates. The printing press, the steam engine, the telegraph, radio, television, computers, the internet, the smartphone in your pocket. None of these required any biological change in the brain. The brain that reads a novel is the same brain that once tracked animal footprints across a savanna. The brain that writes computer code is the same brain that once napped Oldowan choppers from river cobbles. What changed is the cultural environment, the accumulated knowledge, the tools, the institutions, the networks of cooperation that the brain plugs into. And yet, and this is something worth thinking about as you lie there, there are hints that the relationship between culture and biology isn't entirely one-directional. There's evidence that cultural practices can actually drive genetic evolution, creating feedback loops between what we do and what we are. The classic example is lactose tolerance. Most mammals lose the ability to digest lactose after weaning, but in populations that adopted dairy farming. In Europe, in parts of Africa, in the Middle East, a genetic mutation that keeps less lactase production active into adulthood was powerfully selected for. The cultural practice of herding and milking animals created the selection pressure that changed the genome. That happened in just the last 8 to 10,000 years, which is a blink of an eye in evolutionary terms. Could something similar have happened with the brain? Could cultural practices have created selection pressures that shaped cognitive abilities? It's a tantalizing question, and some researchers think the answer is yes. There's a hypothesis, still debated, that the demands of increasingly complex social structures, larger groups, more elaborate hierarchies, more intricate systems of reciprocity and reputation, may have selected for enhanced working memory, better impulse control, more sophisticated theory of mind. The geneticist Bruce Lahn published papers in 2005 identifying genes involved in brain development that appeared to have been under recent positive selection in human populations. Those subsequent studies complicated the picture, and the relationship between those gene variants and actual cognitive differences remains unclear. The honest truth is that we're still in the early days of understanding how culture and genetics co-evolve in the context of the brain. But the principle that what we do can change what we are biologically over enough generations is well established. And there's one more twist to this story that's worth mentioning. Something that might surprise you. Over the last 20 to 30,000 years, the human brain has actually gotten slightly smaller. The average brain volume of a modern human is roughly 1,350 cubic centimeters, compared to about 1,400 to 1,500 in some late Pleistocene populations. That's a decrease of roughly 10%. Now, before you worry about that, there are several possible explanations, and none of them necessarily mean we're getting less intelligent. One idea, proposed by researchers like John Hawkes, is that it's related to a general decrease in body size. As humans became less robust after the end of the Ice Age, brains scaled down proportionally. Another idea is that brains actually became more efficient, that selection favored neural architectures that could do the same work with less tissue, less energy. Think of it like the difference between an early desktop computer that filled half a room and a modern laptop that fits in your bag but has 1,000 times the processing power. And there's a more provocative hypothesis, sometimes called the self-domestication hypothesis, which suggests that as human societies became more cooperative and less tolerant of uncontrolled aggression, the same suite of changes that occurs in domesticated animals, reduced body size, reduced brain size, more juvenile features, occurred in humans too. It's still speculative, but it's the kind of idea that makes you look at the whole arc of brain evolution in a slightly different light. And speaking of looking at the whole arc, that's something worth doing now, because we've come an extraordinarily long way together. From those first simple cells drifting in ancient oceans, to nerve nets pulsing in jellyfish, to the first centralized brains in worms no bigger than your fingernail, through the Cambrian explosion and the rise of vertebrates and the long march onto land, through the age of dinosaurs and the tiny nocturnal mammals hiding in the shadows, through the extinction that cleared the stage and the primates that climbed into the trees and the apes that came back down, through Australopithecus and Homo erectus and the mastery of fire, through Neanderthals and Denisovans and the mosaic genome we carry today, through the emergence of language and symbolic thought and the extraordinary trick of cumulative culture. Through all of that, the brain has been the central character of the story. Not the only character. There's been geology and climate and competition and sheer dumb luck. But the brain has been the thing that kept changing, kept adapting, kept finding new solutions to whatever problem the world threw at it next. And what's remarkable, what's really worth sitting with for a moment, is that there was no plan, no blueprint, no destination. The brain you're using right now to listen to these words, to picture those ancient oceans, to feel that little spark of wonder, that brain was never supposed to exist. It's not the endpoint of some grand design. It's the latest draft in a manuscript that's been revised 4 billion times. And every revision was made, not because someone was steering toward you, but because it happened to work a little better than the version before it, in that particular place, at that particular time. And yet here you are, lying in the dark with 100 billion neurons humming away inside your skull, each one connected to thousands of others. The whole thing consuming about 20 watts of power, roughly the same as a dim light bulb. And with that 20 watts, you can contemplate the origin of the universe. You can remember the smell of your grandmother's kitchen. You can imagine what it would feel like to stand on Mars. You can listen to someone describe a 4 billion year evolutionary journey and feel something about it. That's not nothing. That's extraordinary. Let's talk about where we are now, though, because the brain you have today isn't just a product of evolution in the biological sense. It's a product of everything we've discussed. The genes, yes, but also the culture, the language, the accumulated knowledge of every generation that came before you. And in the last few centuries, and especially the last few decades, something has been happening that's genuinely without precedent in the entire history of life on Earth. We've started building things that think, or at least things that do something that looks an awful lot like thinking. Now, I want to be careful here, because this is a place where it's easy to get carried away, and it's late. And the goal isn't to wind you up with anxiety about artificial intelligence. But it's worth noting, just quietly, that the story of the brain doesn't end with biology anymore. For roughly 520 million years, since those first centralized nervous systems appeared in the Cambrian, the only way to build a brain was the slow, patient, wasteful process of natural selection. Random variation, differential survival, repeat for a few 100 million generations. That was the only game in town. And then, in the last eye blink of evolutionary time, one particular species of primate figured out how to take the principles of computation, which are really just the principles of what neurons do, processing information and making decisions based on inputs, and instantiate them in silicon instead of carbon, in sand, essentially. Melted sand, carefully arranged. The first artificial neural networks were proposed in 1943 by Warren McCulloch and Walter Pitts, a neurophysiologist and a logician working together at the University of Illinois. Their paper was called A Logical Calculus of the Ideas Immanent in Nervous Activity, which is a mouthful, but the core idea was simple and beautiful. They showed that networks of simple on-off units, connected together and following basic logical rules, could in principle compute anything computable. They were directly inspired by what was known about real neurons at the time. The artificial neuron was modeled on the biological one. It receives inputs, sums them up, and fires or doesn't fire based on whether the total exceeds some threshold. Sound familiar? It should. It's a simplified version of what Santiago Ramon y Cajal first sketched over a century ago, what Hodgkin and Huxley formalized in their equations for the squid giant axon, what every neuron in your head is doing right now. But here's the thing, and this is important. Artificial neural networks are inspired by biological brains, but they are not biological brains. They're metaphors made functional. A real neuron is a living cell with thousands of complex molecular processes happening inside it. An artificial neuron is a mathematical function. The gap between them is enormous. And yet, the fact that you can take the basic architectural principle, many simple units connected in networks, learning by adjusting the strength of connections, and make it work in a completely different substrate, in silicon and electricity instead of lipid membranes and sodium ions. That tells you something profound about the principle itself. Suggests that what matters isn't the material. What matters is the pattern, the organization, the way information flows. And that's a very deep idea. One that connects back to something we touched on much earlier when we talked about how the first neurons weren't really invented from scratch. They were assembled from molecular machinery that already existed in single-celled organisms. Ion channels, chemical signaling, electrical excitability. All of it was already there. What evolution did was organize it in a new way. And that's what McCulloch and Pitts did too, in their own way. They took the principle and reorganized it in a new medium. But anyway, the point isn't really about artificial intelligence tonight. The point is about you, about what it means to have the kind of brain you have right now at this particular moment in the history of the universe. Think about what your brain does while you sleep, because that's what's about to happen, or maybe it's already happening. You're drifting. Your eyelids are heavy. Your thoughts are starting to loosen and float, and your brains is not shutting down. Not even close. During sleep, especially during the deep slow-wave stages, your neurons fire in these great synchronized waves, like a stadium crowd doing the wave, rolling across the cortex in slow, rhythmic pulses. And during these waves, something remarkable happens. The experiences of your day, the conversations, the things you read, the problems you were working on, get replayed. The hippocampus, that seahorse-shaped structure tucked deep in the temporal lobe, the one we mentioned when we talked about how mammals evolved the ability to form complex memories, it starts firing in patterns that echo the patterns it recorded during the day. And as it replays them, it's essentially teaching the cortex, transferring memories from short-term storage to long-term storage, consolidating, strengthening the connections that matter, weakening the ones that don't. And there's another process that happens during sleep that we've only recently begun to understand. In 2013, Maiken Nedergaard and her team at the University of Rochester discovered what they called the glymphatic system, essentially a waste clearance system for the brain. During sleep, the spaces between your brain cells expand by about 60%, and cerebrospinal fluid rushes through those expanded channels, flushing out metabolic waste products, including beta amyloid, the protein that accumulates in Alzheimer's disease. Your brain is literally washing itself while you sleep. Cleaning house, taking out the garbage. It's one of the reasons sleep deprivation is so devastating to cognitive function. You're not just missing out on memory consolidation. You're skipping the cleaning cycle. So, as you lie here, as your breathing slows and your muscles relax, and the world outside this moment fades to something soft and distant, know that your brain is about to do some of its most important work. The same organ that evolved from a simple patch of light-sensitive cells on some ancient worm is about to run its nightly maintenance routine, sorting memories, clearing waste, pruning synapses, strengthening the neural pathways that define who you are, and letting the irrelevant ones fade. It's been doing this every night of your life. It did it when you were an infant, when your brain was producing synapses at a rate of over 1 million per second. And it's doing it now, refining and sculpting the network that makes you, you. And here's something I find quietly astonishing. The brain that's doing all of this, your brain right now, contains within it echoes of every stage of the journey we've traced tonight. The brainstem, that ancient stalk at the base of your skull, is running the same basic programs that kept early vertebrate fish alive 400 million years ago. Breathing, heart rate, arousal, the fundamental rhythms of being alive. The cerebellum, that cauliflower-shaped structure at the back, is coordinating your movements using circuits that were refined when our ancestors were navigating three-dimensional forest canopies as early primates. The limbic system, the amygdala, the hippocampus, the hypothalamus, is processing emotion and memory and motivation using architecture that was elaborated during the age of mammals, when being able to remember where food was and who your allies were meant the difference between life and death. And the neocortex, that vast wrinkled sheet draped over everything else, with its 6 layers and its cortical columns, the structure that Vernon Mountcastle's first described. That's where language lives, and abstract thought, and the ability to imagine futures that don't yet exist, and pasts you never witnessed. All of it is there, layered together, not like a neat archaeological dig with clean strata, but more like an old city that's been continuously inhabited for thousands of years. Roman foundations under medieval walls, under modern apartments, all still in use, all interconnected, all part of the same living system. Paul MacLean's old triune brain model, the reptilian brain, the mammalian brain, the human brain stacked on top of each other, was too neat, too clean, and neuroscientists have rightly moved past it. The real picture is messier and more interesting. The old structures didn't just get buried under new ones. They got rewired, repurposed, integrated. Your brainstem talks to your cortex. Your cortex talks to your amygdala. Everything is connected to everything else in ways that are still being mapped. And that messy, interconnected, jury-rigged quality, that's not a flaw. That's the signature of evolution. That's what 4 billion years of tinkering looks like. No engineer would design a brain this way from scratch. It's redundant in some places and fragile in others. It has bottlenecks and workarounds and legacy systems that occasionally cause problems. Like the way your fight-or-flight response, perfectly calibrated for encountering a predator on the savannah, now fires when you get a stressful email. But it works. It works astonishingly well most of the time, for the simple reason that every single one of your ancestors, in an unbroken chain stretching back to the first self-replicating molecules, survived long enough to reproduce. You are the product of 4 billion years of uninterrupted success. Every generation passed the test, and the brains was their most important tool for passing it. So as you drift off tonight, as the slow waves begin to roll across your cortex, and the glymphatic fluid starts its gentle cleaning, and the hippocampus begins its quiet replay of the day, maybe carry with you just this one thought. The fact that you can fall asleep at all, that you can surrender consciousness, let go of the waking world, trust that your brain will keep your heart beating, and your lungs breathing and your memories safe. That's not a small thing. That's the result of hundreds of millions of years of refinement. Sleep itself is ancient. Even fruit flies sleep. Even nematode worms have rest states. The need to periodically withdraw from the world, to let the neural machinery reset and repair, is so fundamental that it predates brains themselves. You're part of that old rhythm now, the same rhythm that pulsed in the first nervous systems, that kept ancient fish safe in their coral crevices at night, that sent early mammals into their burrows when the dinosaurs were still walking overhead. You're joining a tradition that's older than flowers, older than grass, older than birds, and your brain, that improbable, magnificent, messy, beautiful organ, is going to take care of you while you're gone. It knows how to do this. It's been practicing for a very, very long time. So let it do its work. Let the thoughts slow down. Let the details of the day blur and soften. You don't need to hold on to anything right now. The neurons will sort it out. They always do. And tomorrow, when you wake up, the brain that opens your eyes will be the same one that has carried life forward through ice ages and asteroid impacts, and the invention of language and the painting of cave walls, and the building of cities, and the asking of questions that no other organ in the known universe has ever been able to ask. It's yours. It's been yours since before you were born, assembling itself in the dark, following instructions written in a code that's been edited and revised for 4 billion years. And it's still going, still adapting, still learning. Rest now. You've earned it. And so has your brain.